Welcome to your Biocast. I'm Mrs. Yeager and I'll be your guide. As always, take detailed notes and don't forget to bring them to class the next day. Take some time right now to get your binder together, a pen, and get ready to learn more about biology. Welcome to your next Biocast video. This Biocast video is all about RNA, also known as ribonucleic acid. It sounds similar to DNA, and it is similar to DNA in that it does code for the creation of something in your body. RNA specifically codes for the creation of proteins. We'll learn more about that. First, a couple similarities and differences. One similarity that RNA has with DNA is that it's made up of nucleotides, just like DNA is. It's also made up of a sugar, phosphate, and nitrogen base, just like DNA. The major difference between RNA and DNA in their structure is that the sugar in RNA is ribose, and if you remember, the sugar in DNA is deoxyribose. So that's why RNA begins with an R and DNA begins with a D. So RNA ribose. The purpose of RNA, it contains codes for making proteins. Proteins are very important in your body. They're essential for life. Some of these proteins we can get from our diet, we can eat them, and some our body has to make. Okay. So RNA is a single-stranded molecule, which is different from DNA, in that you'll remember DNA is a double-stranded molecule. RNA, here's another major difference, also contains uracil instead of thymine. Okay, so there is no thymine, there will be no A's pairing with T's in RNA, like there is in DNA. Instead, A pairs with U. Adenine is going to pair with uracil. That's one of the major differences. Notice that cytosine still pairs with guanine in RNA, just like it does in DNA. There are three major types of RNA. That's mRNA, which is messenger RNA, tRNA, and rRNA. Those are all the three types. mRNA is the messenger. It carries copies of instructions from the DNA. Okay, so the messenger RNA gets its instructions from the DNA in a process known as transcription, which we'll learn about a little bit later. But just know that mRNA carries messages. rRNA deals with ribosomes. You find these in the ribosomes. If you remember what the function of the ribosome is, it's the organelle in cells that makes proteins, which is why we're talking about it now. tRNA is transfer RNA, and what transfer RNA does is it brings the amino acids over to the ribosome using the message that it got from the mRNA. We'll learn more about this process in a later slide. Okay, this RNA is all about making proteins, but there are two processes that happen. The first is transcription. To transcribe something means to write it, and this process is very similar to writing. This is the process when RNA copies part of the DNA sequence. So first, an enzyme is something that causes a chemical reaction, okay? So an enzyme opens up the DNA, and one strand of the DNA is used as a template to make the messenger RNA. We'll see an example of this later. It says see a drawing on the board, but we'll go over this in a later slide. Just know that in transcription, basically you are writing a copy of the DNA. Okay, and here's the example. So you'll notice that over here we have this strand of DNA that we're starting with. Okay, there's this big blob. This is an enzyme called RNA polymerase. The name of it you don't need to know. You just need to know that it is an enzyme related to the RNA. And what this enzyme does is it breaks apart the two strands of DNA. Okay, and what's going to happen is this RNA here is going to hook up and copy the DNA. Okay, so the mRNA is going to make a strand that is complementary to that DNA. And notice that uracil is only found in RNA. Okay, so we would find uracil on this strand instead of thymine. Okay, so how is the mRNA sequence turned into a protein? Well, first a little bit about proteins. Proteins make up something called amino acids. There are about 20 different amino acids in your body. 10 of those we can get from our diet, and 10 of those our body makes. Amino acids are required to do things like make enzymes, build proteins, create antibodies, and create hormones. They are essential for your cells to function properly. On the left, you'll notice a picture of my bodybuilder friend. She talks often about amino acids because when you get to the level of competitive bodybuilding, you have to make sure that your cells have everything they need to function properly and build muscles. So she takes a lot of supplements containing amino acids. You probably get a lot of your amino acids from your diet. Okay, so how is this code translated? The mRNA code is read 3 letters at a time, and each one of those 3 code sequences is called a codon. So this is a vocabulary word. Codon is a vocab word. And each one of these groups of 3 letters is a codon. So you've got a codon UCG, and you've got a codon AGA. And each one of these codons identifies an amino acid. So the amino acid formed by AGA will probably be different than the amino acid formed by UCG. We'll learn more about that later. Okay, now translating the code. Codons in mRNA give the order that the amino acids should be joined to make a protein. So you're going to get to look up some codons in the chart and determine the amino acid sequence. You don't need to write anything on this slide down. Just go ahead and look at the chart on the next page. So this looks complicated, but it's actually pretty easy to read. Let's start by looking at the codon we're trying to decipher. Let's look at UCG. Here's how you read this chart. You start on the inside and work out. So you look for the section where U is, and we find it here. Okay. Look at what the next letter is. It's C, so that would be here. And the last letter is G. So the amino acid that we create from UCG is serine. Okay. UCA and UCC and UCU also create serine. So some codons code for more than one protein. The second type, let's do another example, AGA. So we start on the inside, again working out. So we've got our first letter, A, and then we'll go to G. Then the last letter's A, so we're going to go right here. So the protein that's produced is arginine. So that's how you read this wheel. It's very simple once you understand it. So now instructions on how to write the RNA strand. The first thing that you need to do is you're going to write the complementary strand of the DNA sequence for the following strand. So you just do what we learned for DNA, and that is how to write a complementary strand. So remember, we're talking about DNA, so A pairs with T and C pairs with G. So your complementary strand would be T, A, C, G, T, A, C, C, A, G, A, T. I hope you see how I'm getting this. And then C, G, T. Okay, so that's your complementary strand. And the next step is to write the sequence that would be transcribed from the complementary DNA. So all you would do is pair it up just like we did there, but remembering that, that A now pairs with U. Okay, so we're going to treat these T's as though they were U's. Okay, so T is going to pair, we'll treat it like it's a U, it'll pair with A. A will pair with U. C C will pair with G, that stays the same, okay? And now we're working on GTA right here. So G pairs with C, T, we'll treat that like it's a U, pairs with A, and T, I'm sorry, I made a mistake. In between here, A is going to pair with U. Okay, so those are the first two strands, AUG and CUA. So you could continue out. Remember, each one of these three groups is called a codon. Okay, so we're going to look up and see what the codon AUG stands for, which amino acid. So let's go to the chart, and we're looking for AUG. So sorry about that. So we start from the inside again, AUG. So this particular codon codes for methionine. The second part of the protein synthesis process is translation, and this is the actual production of proteins. Remember that this happens inside the ribosomes in a cell. The ribosomes are those organelles that create proteins. And translation begins when the messenger RNA attaches to a ribosome. So the mRNA will hook up with the ribosome, and then the amino acids are carried to the ribosome by transfer RNA. So you could say that the tRNA transfers amino acids to the ribosome. And here's kind of what that process looks like. No need to draw this down, I just want you to take a look at it. Okay, so here's what's happening. We have inside the nucleus of the cell, this mRNA is being created. Okay, this is the messenger. And the messenger RNA, this is the same strand shown down here, so don't get confused. The messenger RNA hooks up with a ribosome. This ribosome is this big structure here, okay? That's that organelle that floats around in the cytoplasm and produces proteins. Then the transfer RNA will read, the ribosome reads this mRNA and says, okay, we have AUG, we need a complementary strand, which would be UAC. So then what happens is this transfer RNA will basically go and get those nucleotides that the mRNA needs and hook up with them, okay? And in the process, proteins are created. Here's your challenge, your at-home challenge. I want you to write the RNA sequence that would be transcribed from the following DNA strand. Okay, so the first thing that you need to do is write down this strand right here. Okay, this is your strand of DNA, and the first thing you need to do, when you're done with that, you're going to write the complementary strand. So to get you started, C pairs with G, T pairs with A. Remember, we're writing the DNA strand first. A pairs with T, and you can continue. And from there, then you will write the RNA strand, and remember that A pairs with U, okay? So go ahead and give that a try. If that's confusing to you, I would go back a few slides and look at the other example that we did. Make sure that you bring your notes to class with you, and I will see you for your next Biocast.